Okay, folks, let's dive back into it. I just remembered as I'm going through this, well, first of all, I apologize. We just discovered that the camera was pointed sort of halfway down here, so the video for this is going to be odd. I may just use the video. If the capture didn't work yesterday, so I may make the video for today's just be the capture, since it should have everything on there. You don't need to see my smiling face, I think, all the time. So when I get the links working for both of these, you can watch the capture, which will show only the screen, which has the advantage it'll probably be a little higher quality video, which is why I'm using this. I'm not using a laser pointer. And the other videos that'll be on YouTube will be the regular camera. And I'm using the camera as a backup because, like yesterday, the screen capture didn't work, and so I'm glad I had it. Today, the screen capture may back up this. So the screen capture will have my voice, but I forgot to put this on for the first couple minutes. So today's a total screw up. So okay, so. Now, the other thing that I remembered I forgot to tell you was when we talk about amino acids, there's a term I didn't give you, and I need to give you that. So when we talk about amino acids, all right, remember I had an amino acid and I had a charge of -1. I had a charge of +1, then I lost a protein, had a charge of 0, and then I lost a protein, had a charge of -1. The place where an amino acid has a charge of 0, it's known as a zwitterion. Z-W-I-T-T-E-R-I-O-N. Okay? A zwitterion is an amino acid or any molecule that has an overall net charge of 0. Okay? Now, I haven't gone through and shown you a calculation of PI. That's something I've talked to Anna about, and she's going to do that for you in recitation, probably next time, but it may not happen until next week, depending on how far she got today. The other thing about PI is if you look at my videos online, you'll see me showing you how to calculate PI. PI has to be calculated. It cannot be estimated. It has to be calculated. And it's calculated based on pKa values, and you'll see how that's done if you look at that video. So I'll leave you with that. Okay. Well, I'm talking about beta strands and arranged into beta sheets. And here's some representations of some beta strands into different sheet forms. We can imagine that here's a protein that has a beta strand. It goes down here. It loops up. It loops up. We can have different ones in different parts of the protein that are oriented in opposite ways. So this one might go for a ways and curl around over here, come up here. This guy might come around, then come back up here, etc. So the orientation is simply showing you the amino to carboxyl direction within a given polypeptide chain. It's not really important for our purposes, but you can see some really beautiful structures with these. Some of these things are arranged in barrels, for example. They call them beta barrels. And the barrels have important functions in the confines of an enzyme. One of the things that barrels do is they help sequester things. So if you have a molecule, for example, that is unstable in water, there's a famous enzyme that actually catalyzes two reactions. It catalyzes one reaction up here, and the product is unstable in water. So that rather than release it into water where it would fall apart, it moves it through the barrel to another part of itself where it catalyzes the second reaction. So the molecule never gets a chance to see water. And so structure, as you can imagine in a case like that, is very important to function. If the barrel isn't there, as soon as that first molecule made it falls apart in water and the cell is hosed. All right. Okay, let's see. There's a barrel. This is a part of an enzyme that makes fatty acids. And fatty acids are fairly nonpolar things. They don't like being in water. And you can imagine that that barrel is involved in helping to protect that fatty acid from water that it doesn't like to associate with. This structure also shows us a couple of other things. It shows us, first of all, that proteins have more than one structure. That guy up there, it's a tiny alpha helix. These guys down here, these are beta strands. And these things, these yellow coils in the middle, are sort of connectors of the individual beta strands. They're part of the polypeptide chain, but you'll notice that they don't have any regular structure. They have no repeat. Remember, the definition of secondary structure was something that had a repeat. These guys have an odd name. They're called random coils. And they're not really random, but we call them random coils because they're very difficult to predict exactly what configuration they will have. So random coils turn out to be important, and we can, for our purposes right now, think of them in simple terms as bends. Think of them in simple terms as bends. Okay? Here are some things that will make bends. Okay? What you see on the screen is a bend. It's a sequence of 4 amino acids, I plus 1, I plus 2, and I plus 3. Okay? And these actually have a hydrogen bond that's helping to stabilize it. But bends happen, be a good bumper sticker, right? Bends happen, wouldn't be a very good one for a skin diver though, right? Bends happen, all right, because there's something that won't let it go into other structures. One of the common amino acids we find in bends, yes, you guessed it, it's your friend proline. Proline is commonly found in bend structures. And another amino acid that we commonly find in bend structures is glycine. Now, glycine was the simplest amino acid. Why do you suppose glycine would be found in bends?
Because proline is so big.
It can help proline, that's one. Because of its flexibility, it can actually allow other things to form. That's part of it. And the other is the small size of the R group. The very small size of the R group allows for all kinds of possibilities, and it's not really dictating anything. It's a pretty flexible amino acid and allows for bends to occur where a protein really needs to bend. Okay. There's another sort of structure of some beta strands arranged in a sort of a barrel. Okay. And next I want to talk about some proteins that have, is this thing still on? Yeah. Some proteins that have almost only primary and secondary structure. Most proteins have primary, secondary, and tertiary structure, but not all proteins. Some proteins have only primary and essentially secondary. That's basically what they have. These proteins are different. They are not uncommon, as I will explain to you, but they're different. So here's an example of a protein that is what we describe as a coil of coils. We see an alpha helix that's wound with another alpha helix. Okay? Notice that this alpha helix seems to, both alpha helices seem to go on forever and ever and ever. There's no bends. So there's the first thing we notice about these fibrous proteins, no bends. They're just going on and on and on and on. Just like a fiber will go on and on and on and on. Kind of like my voice going on and on and on and on. Okay? These guys, examples of fibrous proteins, your hair. Your hair is a real good example of a fibrous protein. And you think, well, my hair has a three-dimensionality to it. My hair does have curls, for example. Okay? So how do curls arise? Well, curls arise because of some little bitty bends. Okay? So there's nothing absolute about these. And these little bitty bends happen as a result of disulfide bonds. So when I said that two sulfurs could react with each other, if I have a sulfur, let's say, right here, that has a sulfhydryl group sticking out, and I have another sulfur right here that has a sulfhydryl group sticking out, and these guys make a little bend, and they bind to each other, it's going to cause a little kink at that point. That's what curls your hair, disulfide bonds. In the old days when people used to go and get permanents at the beauty shop, I never did, but in the old days when people used to go and do that, if you ever know anybody who goes and does that, they go and they come out and their hair smells like crap. Right? The reason that their hair smells like crap is to break disulfide bonds, you have to use an agent that's a sulfur agent, kind of like the stuff that's there, and sulfur stinks. Permanents aren't permanent. They don't stay there forever, but to change the curl or give curl to hair, you have to break and remake disulfide bonds, and that's what that stinky reagent is that they're doing, putting on hair. Eventually those will break themselves and go back to a more regular, natural form, but that's what happens in making a permanent. Okay, so hair is an example of a fibrous protein. Okay, here's a very interesting, another example of a fibrous protein. Silk. Silk is mostly beta strands organized in sheets. That's what silk is. Okay. Silk is made from a protein called fibrin, and it's mostly beta strands arranged in sheets. Now, these fibrous proteins, as I said, are mostly primary and secondary structure. Virtually no bends, virtually no other considerations that they have. Okay? Essentially primary and secondary structure. What I have on the screen is something that is a sequence of the most abundant protein you have in your body. It's collagen. And collagen is a real oddball in this scheme because it's going to break the rules of everything that I told you. I told you that proline really doesn't like to be in a helix. I told you that proline was very unflexible. And now I'm going, if you look at the screen, you see every place you see a pro, that's proline, HYP, that's a modified form of proline called hydroxyproline. This sucker is a fibrous protein that's full of prolines. Basically, Kevin's been lying to you. Okay? Well, not quite, of course. If we look at this, there's something else that strikes us about this sequence besides the prolines. What is it? Glycine. And what property did glycine have? Glycine had virtually no steric hindrance, and glycine permitted a lot of other things to happen. So it didn't have to go into a turn. It turns out that we can actually take proline. If we have appropriate numbers and positions of glycines, we can actually make it into helices. And that's actually what collagen does. Collagen is literally the glue that holds us together. Okay? It's literally the glue that holds us together. Okay? This shows the arrangement, glycine, proline, proline, glycine, proline, proline, okay, giving us flexibility and allowing for the formation of helices that would otherwise not be able to form. Collagen is a real good example of coiled helices. All right? 3 of them are interwoven together. Now, I've got a story to tell you about collagen that I think will hopefully teach you something important about nutrition. Okay? I'm trying to see, does anybody here have braided hair? Nobody has braided hair. You guys have braided your hair. Okay? When you braid your hair, you do something important with it. You take 3 strands and you intertwine them, and then you tie it off with a rubber band at the bottom. Because if you don't tie it off with a rubber band, what happens to your braid? It comes undone and you don't have a braid for very long, right? Okay? Well, keeping those strands together is a very important thing. Collagen has a braid. It's a braid of 3 strands, and strengthening those strands are covalent bonds between the strands. So instead of putting an external rubber band onto it, there are bonds that are made between the strands. So there are places where this red guy will form a covalent bond with the blue guy, the red guy with the yellow, the blue guy with the yellow, etc. By holding the strands together, collagen gains strength, just like a rope gains strength when we braid it. We take 3 ropes together and we braid them together. We've got a stronger rope than any individual rope would be, right? Okay. Well, how does that relate to nutrition? It relates to nutrition as follows. Oh, I did it, didn't I? Oh, there we go. Okay. It relates as follows. Let's go back to that sequence. We look at the sequence, and here was this hydroxyproline. You hadn't heard about hydroxyproline before. What the heck is hydroxyproline? Hydroxyproline is an example of what we describe as post-translational modification. Translation is the process of making a protein. So post-translational modification means after the protein is made, something is modified. In this case, the modification is that proline is gaining hydroxyl groups. Something is chemically putting hydroxyl groups onto those prolines. And in the process, creating this unusual amino acid called hydroxyproline. This is about the only place we find it. It's those hydroxyl groups that are forming bonds between the strands, splitting out water and causing a covalent bond to be formed. So these guys are being put onto the prolines of collagen to strengthen the collagen. If you can't put the hydroxyl groups onto the proline, you can't have and you won't have strong collagen. Your collagen will literally fall apart. Well, if collagen is the glue that holds you together and your collagen is weak, you're in bad shape. Okay? Now we come to the nutrition part. How do those groups get put onto there? Well, it turns out those groups get put onto there as a result of catalysis that involves vitamin C. Okay? Vitamin C is necessary to put those hydroxyl groups onto proline to make hydroxyproline. If you are deficient in vitamin C, you will not be able to do that. You will literally fall apart. You develop a condition called scurvy. Now, the way that this disease was actually discovered was back in the old days when they had pirates and people who lived on the sea. They ate salty meat because they didn't have refrigeration, and salt was the way that they could keep things, and they didn't exactly have a little brie and arugula for part of what they were eating. They didn't have vegetables on the ship. Vegetables are where we get a lot of our, and fruits where we get a lot of our vitamin C. These big honking pirates would get out on the wild ocean and be gone for a year, and they would come back and they would be little wimpy guys because they've all developed scurvy because they don't have vitamin C. Okay? And thanks to vitamin C, they're actually able to have strong collagen and be nice, healthy people. We don't see scurvy today because we have enough vitamin C in our diet that we don't have that happen. Okay? Another thing happens with your collagen is that the older you get, the more you retain collagen that's got those bonds in it. The older you get, the less flexible you get. And let me tell you, it's true. I got too much old collagen in me. Those bonds, if I could just get them to let go a little scurvy wouldn't be bad. Right? Want to have a little bit more flexibility. But the reason you become less flexible the older you get is you retain more of that cross-linked collagen. So my collagen is stronger than yours, okay, but less flexible as a consequence. Okay. I've got to get used to that. I just haven't gotten used to it. All right. And let's see, what's that? Okay, blah, blah, blah. Okay. Now, that's basically what I want to say with respect to secondary structure. We can now turn our attention to tertiary structure. We're moving through this nicely, actually. Okay? We finished 2 levels of protein structure. The first level told us that amino acid sequence was important. The second structure told us that at a local level, we can have some repeating structures, alpha helix, beta strands. And I talked about random coils. The question always comes up, well, is a random coil secondary structure or tertiary structure? And it's really a semantic argument. If you take the argument that the repeating structure has to be in place, then it's not a secondary structure. Okay? So you can call it a tertiary structure if you want. Some people classify it as a secondary structure. Some people classify it as a tertiary. For my purposes, if you know what a random coil is, I'm happy. Okay? I won't count you wrong for a random coil being a secondary or a tertiary. But knowing that a random coil is somewhere in between the two, just based on a semantic argument. Tertiary structure, as we will see, however, is very, very important. Tertiary structure arises from those random coils. That's where we get our tertiary structures from. Okay? That's where we get those tertiary structures from. So let's take a look at some of those. And the first one we'll talk about is in a protein we'll say, well, several things about this term. It's very important. And it's a protein called myoglobin. Now, myoglobin, of course, sounds like hemoglobin. And the reason it does is because it's structurally very similar to hemoglobin. Myoglobin is an oxygen-storing protein that we have in our muscles. That's where the myo part comes from. Okay? Myoglobin has a primary structure. You can see that it has several secondary structures. There's an alpha helix, 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 there's an alpha helix. A lot of alpha helices there. You don't see any beta strands in this guy, for example. Most proteins, by the way, have a sort of mixture of the two. They don't have just one or the other. They have a sort of mix of the two. This guy doesn't have any beta strands. It's got alpha helix, and it's got turns. And those turns give this guy a three-dimensional configuration. Right? This protein has done something that we call folding. It has folded. We can think about this protein, if we laid it out on the table, we would see it lay out as a nice linear piece. But that protein, as it's being made, doesn't come out linearly. It starts folding up. It starts to assume this shape, in many cases, as it's being made. Okay? The folds are arising from those random coils. That's where the folding's coming from. Folding is coming from that. Now, this guy also has something else in here that we haven't seen before. It has something called a prosthetic group. And all that a prosthetic group is another group that's not an amino acid that helps a protein to do something. So it's a group, it's basically a molecular structure that's not an amino acid that helps a protein to do something. We'll see several examples of this. But this prosthetic group is known as heme. It's the same heme that gives hemoglobin its name, but since the name hemoglobin was taken, this guy got called myoglobin. Now, the heme group is important, as we shall see. The heme group, I hope to convince you, probably in the lecture, I think it's tomorrow or the next day, is one of the most fascinating molecules in our body. But this guy has in the very middle of it an iron atom, and that iron atom holds oxygen. Okay? I said myoglobin was a protein that's very good at storing oxygen. And that's its primary function in the muscles. It stores oxygen, and we'll see what it does with it later. This guy has a distinctive three-dimensional shape. If I destroy this guy's distinctive three-dimensional shape, I will destroy its function. Again, structure is necessary for function. If I heat this guy up to a high temperature, it's going to lose its shape. It's going to lose its function. Okay? Very important consideration. What we see on the left is what's called a ribbon diagram. It's simply to show our eyes the shapes that are there, because those shapes are easier to see than they are to see in a molecular packing model, which is what we have over here. Okay? This molecular packing model, it's a little harder to see those shapes. The ribbon diagram gives us a very nice depiction of those. But they're just different ways of representing the same thing. Okay? Now, my purpose in showing you this is to plant in your head that this guy has a distinctive shape. Okay? And that distinctive shape arose because of folding. And the question is, yes, Anesia?
My question was, is the heme group attached to the R groups within the protein, or is it?
Okay, very good question. So the heme group is, in fact, attached to the R group side chains of the protein. They are. And it turns out that the heme is attached to the side chains of histidines. Yep, it's covalently attached to them, but it's a very good question. We'll actually see that later on. Okay, now, it has a distinctive shape. The distinctive shape arose because of folding. Well, what caused folding to happen? There's my pop quiz for you today. What causes folding to happen?
Random coils?
Random coils are part of the folding process, but what causes random coils to form? And where do R groups come from? Amino acids, but you're getting to the core, and we haven't gotten to the core yet. Steric hindrance arises because of structure, right? Where did the structure come from? Primary sequence, exactly. Every property of a protein comes from primary sequence. So ultimately, this three-dimensional shape came about from primary sequence. The secondary structure came about from primary structure. The sequence of amino acids dictated this was going to be an alpha helix, because if there were certain amino acids in this early part of the protein, that wouldn't be an alpha helix. It couldn't exist because of steric hindrance. The sequence determined that. The sequence is dictating everything. So ultimately, the structure that I have for this protein is a function of the sequence of amino acids in there. That's why storing that information in the genetic code is so important, because the genetic code tells our ribosomes the sequences of amino acids to make, and when those sequences of amino acids come together, they form proteins with the right shapes that have the right functions. Those are very, very important things. But all of it ultimately is traced to the sequence. If I change the sequence, that's the other thing that can happen. If I change the sequence, I may change the function. We all have heard of mutation. Mutation changes the DNA codes for the protein. Some mutations, if I put a mutation of an amino acid right here that would disrupt that alpha helix, I'm going to disrupt the structure of this protein. I might lose the function of this protein. I might make this function of this protein be even better. And you think, does that ever happen? And I say, if that didn't happen, you wouldn't be sitting here today. That's the driving force of evolution. Okay? So mutation also, over evolutionary time, affects how these things even come about and how they exist in the first place. Okay, so tertiary structure. I'm going to define it for you now. I've told you a lot about it. I'm going to define tertiary structure for you. I think it logically follows, but I'm going to describe it. Tertiary structure is the three-dimensional structure of a protein that arises from interactions between amino acids that are not close in primary sequence. I'll say it again. Tertiary structure is the three-dimensional structure of a protein that arises from interactions between amino acids that are not, underline not, close in primary structure. Now, what does that last part mean? Secondary structure, we said it arose between amino acids that were close, so we said this might be number 4, this might be number 10, they're close. What does not close mean? Not close means more than 10 apart. And on this structure, what does it mean? It means this. Look at how this helix is close to this helix. How did they get close to each other? The answer, folding. But this helix might be amino acid number 121, and this might be amino acid number 17. They've been brought into close proximity as a result of the folding process. And interactions between them help to stabilize this structure. We're going to talk about those interactions in a second. But interactions between those are going to stabilize this structure. Yes, sir?
So the beta barrel and beta sheets be tertiary structure then?
Well, beta barrels and beta sheets be tertiary structure. They can give rise to tertiary structures, but technically the beta strands themselves, that's why I call them strands, are secondary structures. Beta barrels are what people describe as super secondary structure. It's a cop-out.
Okay.
Okay? But it's a very good question. Personally, I say, yes, they're tertiary structures.
So for this class, yes they are?
For this class, you can call them a tertiary structure if you wish. How's that?
Okay, thanks.
Okay. All right. Now, so we see interactions can happen between amino acids that are not close in primary sequence. And whenever we have a structure, the reason we have a structure is because that structure has some stability. There have to be things that stabilize structures. Interactions stabilize structures. Well, let's think about the kinds of interactions that we can have. One kind of interaction that you've already talked about, what interaction would we expect might be between two things here? Hydrogen bonds, right? Yeah, hydrogen bonds are everywhere, okay? Hydrogen bonds are one of the forces that can stabilize a tertiary structure. There's another one, okay? Imagine that I had, for example, a plus charge here and a minus charge over there. They would be attracted to each other. That could help to stabilize the structure. And so we could say that ionic forces or ionic bonds could help to stabilize a tertiary structure. Not quite so easy to do that in secondary because it's kind of hard to get them in close proximity. But in a tertiary, yes. Okay? Disulfide bonds. What if I had a sulfur that reacted with a sulfur over here and formed a covalent bond between them? Yes, absolutely. Disulfide bonds, in fact, turn out to be the strongest forces that stabilize tertiary structure. Covalent bonds are hard to break. And a sulfur-sulfur bond is not an easy bond to break. So once it forms, it gives some structural integrity to that protein. Okay? There's another category of bond that I really won't talk about here. It's called a metallic bond. And some metals will, in fact, help to stabilize structures in proteins. They're not really relevant for our purposes, but they are one of the categories of stabilization. Now, the last category of stabilization that I want to talk about that exists in tertiary structure is one that I've briefly mentioned before, but I haven't shown it to you. Come on. I haven't shown it to you, and I want to do that, okay? And I thought I had it on here. I guess I don't have it on here. Okay. Go back to this. Okay. And it's a bond we call hydrophobic bond. Now, I like to think about it, it's described as a hydrophobic bond. I like to think of it more as a hydrophobic attraction. It's a hydrophobic interaction. What does that mean? Well, if you think back to the aliphatic amino acids that I described to you, I said that they didn't like water. They didn't like water very much. And so if we have molecules that don't like water, let's say we take oil and we mix it with water and we shake it up, eventually it will separate out into two layers. The oil that doesn't like water associates with itself. The water that doesn't like the oil associates with itself, and that's why we get two layers. In proteins, similar things happen. There are portions of a protein that really don't like water. And they want to associate with each other. And so they actually do associate with each other, and that's something that helps the folding process to happen. They avoid water. They associate with each other just like a group of nerds at a party that don't want to talk to anybody else. You see them over in the corner, right? They're over there talking to themselves, and no, don't look at me, you know, right? Okay? The nerds in the proteins are the hydrophobic amino acids that are associating with each other. They're not associating with anybody else, and they fold and they stay on the inside of the protein. That's what a hydrophobic interaction does. So when we look at the structure of most proteins that are dissolved in water, what we discover is that on the inside we find amino acids that have side chains that don't like water, like the aliphatic ones. And on the outside, we find a preference for amino acids that do like water, the hydrophilic ones, like serine, like threonine, like aspartic acid, like lysine. The ones that have charge, the ones that have hydroxides and so forth, they like to be on the outside. Those that don't like to interact with water tend to be on the inside. Those forces I just described to you are the forces that stabilize tertiary bonds. Tertiary structure, sorry, using the wrong words. Okay. Now, I mentioned disulfide bonds. Here's a disulfide bond. Okay. Here's a cysteine. Here's a folded protein that's brought it into close proximity with another cysteine. This might be amino acid number 41. This might be amino acid number 216. But in the folded structure, they've been brought into close proximity. Voila, they like to react. When they react, it's actually an oxidation. Electrons and protons are given up, as you see here. The resulting bond is called a disulfide bond. That disulfide bond is very strong. As I said, it's the strongest force that stabilizes tertiary structure. It can be broken chemically fairly easily. It involves reduction. And that's what's happening in that permanent that's going on the lady's hair, or the guy's hair, as the case may be. Okay? Anesia?
Is cysteine the only amino acid that forms a disulfide?
Cysteine is the only one that forms disulfide bonds. That's right. There's only one other amino acid that has sulfur in it. It's methionine, and it doesn't have a sulfhydryl. It actually takes a sulfhydryl in order to form a disulfide bond. Okay? Okay. Now, let's see. What else can I tell you? So we can break disulfide bonds. I said we can break them chemically. There are chemicals that will do that. There's sulfur-containing compounds. A very common one that we see is called mercaptoethanol. I don't know if you've ever worked with mercaptoethanol. Anybody ever worked with mercaptoethanol in the lab? Nobody? Okay, it smells kind of like rotten eggs. And a little tiny drop of it can fill an entire room. One of the worst things that you can do when you're starting out working in a laboratory as a student is break the bottle that has mercaptoethanol in it. Because I mean to tell you, you can evacuate a building with this stuff. It is really foul stuff. It'll knock you over, okay? Well, what mercaptoethanol does is it donates protons and electrons to this guy and drives this reaction back this direction so that we make these two here. We've just destabilized a protein in doing that. Destabilized a protein in doing that. Okay. By the way, I don't use this term cysteine. I don't think it makes any sense. I just call this a disulfide, or two cysteines that are linked together. Cysteine doesn't make any sense to me. Gesundheit. Okay, now, I've gone to all this trouble to teach you about tertiary structure, and you've got it all figured out because now you know, for example, oh, come on. What? Oh, there's the problem. You know, for example, that the hydrophobic amino acids are found on the inside. And now I'm going to break my own rule. I'm pretty good at making rules and then turning around and breaking them. Well, this is an unusual rule, and I think it'll actually teach you something about protein structure. Okay? This is an interesting protein called porin. Okay? And porin is a protein that we find not dissolved in the water of the cytoplasm, but rather we find it embedded in the lipid bilayer of the cell membrane. The function of porin is to let in water. Porin meaning it's like a pore that has an opening that lets water in. So it's got a little hole, all right? What we see on this guy here is the structure of porin with the relative hydrophobic amino acids and hydrophilic amino acids colored. And if we look at it, it's very odd. The hydrophobics are on the outside, and at least some of the hydrophobics being the yellow ones, and at least some of the hydrophilics, the blues, are on the inside. This guy is inside out. What gives? That broke our rule. Well, you'll notice when I gave you the rule, I said it was for most proteins that are dissolved in water. This guy is not dissolved in water. This guy is embedded in a lipid bilayer. A lipid bilayer has on the outside polar parts, and on the inside it has nonpolar fatty acids. This protein exists. It's buried in this nonpolar layer of fatty acids. And guess what? If you want to associate with nonpolar fatty acids, you darn sure better have some hydrophobics that are out there to associate with them. So because this guy is embedded in a membrane, it's sort of inside out. The hydrophobics are on the outside to interact with the fatty acids of the membrane. The inside is hydrophilic because, hey, that's where the water's going through. Water has got to interact with the inside portion of this guy. So porin is a rare example of inversion of that rule that I gave you. If I give you a protein that's dissolved in water, you may assume it has that rule I gave you. That is, hydrophobics inside, away from water, hydrophilics outside, associating with water. Okay. I keep doing that. Why don't I learn that? Stupid professor. All right. Well, we're doing very well here. Other questions? Yeah.
I just said it was all your collagen.
What's that? It's all my collagen. It's just going. I'll tell you, by the end of this lecture, my collagen feels like it's going away. Nothing much more about that. All right, we're actually in pretty good shape. Let me finish tertiary structure, and then I thought we might do another song. How's that? Okay. All right. So we have something to look forward to. Something besides, well, maybe my singing isn't something to look forward to, but we'll see. All right. We'll talk later in the term about insulin. Insulin is a fascinating protein, and it's a protein that begins to show us something else about protein structure. Now, we'll see that something else when we talk about quaternary structure in just a bit, but this is our first hint of quaternary structure. Insulin turns out to be a fairly short polypeptide. Insulin is a protein hormone. Our body makes insulin after we have a meal, and the reason our body makes insulin after we have a meal is because insulin stimulates our cells to take up glucose, because in our bloodstream, glucose is a poison. We'll talk about that later. But insulin is stimulated by is made by our body to stimulate our cells to take glucose out of the bloodstream so we don't poison ourselves. You think I'm joking? I'll give you some evidence for it later. Okay? Now, this guy, as I said, is a protein hormone, meaning it's a protein that acts as a hormone. And a hormone is something, you don't need to know this definition, but a hormone is something that travels in our bloodstream, communicating different parts of the body. Our pancreas releases insulin to the rest of our body to communicate, hey, blood glucose levels are getting high, you're going to poison yourself, we better get this glucose out of the bloodstream. Okay? This insulin shows us, okay, some of the power of disulfide bonds. Look at this. This guy has an A chain and it has a B chain. The two chains are held together only by disulfide bonds. Now, it turns out insulin is made originally as a single polypeptide chain. Originally, when this guy is made, this loop right here, this guy over here has a loop of a bunch of amino acids that come around here and then are connected over here. And then a protease, which is an enzyme that breaks down proteins, a protease comes along and clips off a piece, and you're left with two single polypeptide chains. Now, my question to you is, if we're going to get insulin, what has to happen first before the protease comes along? Something important, darn sure had better have happened first, or we're not going to have functional insulin. What had to happen first? What's that? Sequence is put together, but what else had to happen? Folding. Excellent. Okay. Folding had to happen. How do we know folding had to happen? It's right on the screen. Disulfide bonds had to form, and they had to form in specific places, right? We got a lot of places where disulfide bonds could form. What if this sulfur from amino acid 21 linked over here with amino acid number 7? We wouldn't have the same structure, right? Folding dictated that this guy was only going to form a disulfide with this one, and this disulfide over here was only going to form a disulfide with this one, and this guy was only going to form a disulfide with this one. We didn't have random association of those, and that meant that folding had to happen first. Very good. So folding had to happen in order for us to get this exact structure. This structure starts to introduce us to the last important component about protein structure, and the last important component is called quaternary structure. I'm not going to talk about it today, but you can actually see it on the screen. Quaternary structure arises when different polypeptide chains interact with each other. This guy started out as one chain, but right now they're two separate chains. Two separate chains, and these two separate chains are interacting. They're actually being stabilized by disulfide bonds. Not all quaternary structure is stabilized by disulfide bonds, but some of them are, and here's an example of one that is. Okay, so that is what happens there. Let's see, what do I have here? I'm not going to worry about that, and I'm not going to worry about that. Okay, I think we're at a point we could actually do a song. Let's do a song, and then we'll call it a day. How's that? The song I have for you today is all about protein structure. It's to the tune of "Oh Christmas Tree." And let's go here. Oh, I'm sorry. I got the wrong one. It's Oh Little Town of Bethlehem. That's a different one. Sorry. Okay. It says instructor sings and everyone sings. Let's just everybody sing the whole thing. So, oh little protein molecule, you're lovely and serene, with 20 Zwitter ions like cysteine and alanine. Your secondary structure has pitches and repeats, can't hear you, arranged in alpha helices and beta pleated sheets. The Ramachandran plots are predictions made to try to tell the structures you can have for angles phi and psi. And tertiary structure gives polypeptide zing because of magic that occurs in protein folding. A folded enzyme's active and starts to catalyze when activators bind into its allosteric sites. We'll talk about that later. Some other mechanisms control the enzyme rates by regulating synthesis and placement of phosphates. And all the regulation that's found inside of cells reminds the students learning it of pathways straight from hell. So here's how to remember the phosphate strategies. They turn the GPBs to A's and GSAs to B's. We'll talk about that later too. So, okay, let's call it a day, folks. It's a warm one.
Oh, yeah.
Thank you. See you tomorrow.
Yeah.
See you guys tomorrow.
Same bad time, same bad time.
Same bad time. Amen. Stay cool.
Yeah.
Ooh, it feels cool back here. That window's nice.